Assalamu alaikum. Hello everyone. My name is Dr. Farhan. I am teaching Bioinformatics course to undergraduates and postgraduate students. This is the course I've been teaching from last couple of months. So I started with the basic concepts of biology and then bioinformatics, why bioinformatics is important, different types of databases and what types of information are available on databases. And then from that we went into sequence alignment, and from sequence alignment we did multiple sequence alignment, and from multiple sequence alignment we performed phylogenetics, one of the applications of bioinformatics. All these lectures are available on my channel, so if you want to go through the whole process, you can start from my students are they have the obligation to do it, but if any other person want to watch these lectures, so he should start from the bioinformatics section. So today's topic is disease informatics. This is the very important application of bioinformatics, and these days, there is a term which is coined like data science, big data, right? And everybody is very maybe some are excited and some are afraid of this term, that big data. That why I'm a biologist, I don't know how to perform the analysis, how I can cope with the big data. I cannot even cope with some small information. So these sort of challenges and exciting aspects are there, and but the bioinformatics is playing its role in making things simpler for the biologists as well, and coming up with the new challenges for the bioinformaticians or the data scientists. So this is a very integrative process, and everybody has its role to play. Now in 2020, the majority of the biologists have realized that without dealing with the big data, they are nowhere. So on the other hand, bioinformatics scientists have provided a lot of solutions to the biologists so that they can analyze the big data using some easiest approaches. So let's start from the beginning, that how bioinformatics or disease informatics is important, and why data science or big data is very important for the students of biology. Why? So this is one figure from the Michael Stratton, one of the very prominent scientists, and it was published way back 10 years ago, 11 years ago, and it tells you that why big data is important. That now we know that everything starts at the DNA level, right? And DNA is the important part, and that this DNA starts from the very basic fertilized cell, and it moves towards the gestation, infancy, child. For example, here we are talking about any disease or for example, here we are talking about cancer, right? So there's a series of changes that occur throughout the process, starting from the fertilized egg to the adulthood, and from adulthood to other. Let's start one by one. That, for example, here you can see that if there's a mutation, first mutation was occurred at this gestation level, and that multiplied at the infancy into 2, and then from infancy there are some other types of changes. For instance, this yellow represents that intrinsic mutation processes, this blue represents that these are the environmental or lifestyle exposures, and then there are mutations like other mutations like star represents that these are the driver mutations. So we know that there's a difference between a passenger mutation and a driver mutation. It's very simple. Now you know that this as a driver actually drives the cell, right? So driver means that the mutation which drives the tumor, and passenger is the mutation which actually goes with the tumor. It is not actually the driving factor, right? So there are different types of mutations that start from the first day till from the fertilized egg to the gestation, then infancy, then childhood, and then adulthood, and this expands to a big number, and then this expansion leads to the tumor, and the tumor can then metastasize or get invasive to late stages, and then metastasis, and then that becomes resistant to the chemotherapy or any drug or anything you want to give, or we want to cure the disease. Now, with the passage of time, it becomes very challenging for all the for the individual as well as for the biologist to identify and to learn and to analyze all these mutations. And these mutations actually are in very big numbers. So dealing with big number or big data is the real challenge these days. So there are a lot of different concepts which you should go through, and you should know that you can analyze them one by one to understand the data types in disease informatics. For example, here one type is the mutation. So these are mutations. Then there are other chemotherapy resistant mutations. So there can be a difference between the passenger mutations and the chemotherapy resistance. So you know these. There are three types of three data types of data mentioned in this box. So this data increases, and the increase in data makes things complicated. For example, these changes can be of multiple types. Here you can see that it's written in the single nucleotide polymorphisms and or point mutations. You know the difference between single nucleotide polymorphisms and point mutations, right? And then insertion. What is the difference? Single nucleotide polymorphisms or substitutions can and the mutation is different in a way that mutation can actually change can always or possibly be produce a drastic change on the gene. However, single nucleotide polymorphisms are present in more than 1% of the population, and it can be neutral substitution, and it occurs throughout the life in all the individuals which do not give any phenotype. So, and then there are insertions, and this is 1 point change, right? One point. Then there are insertions and deletions that can be to start from 2 to 3, 2 to 10 base pair change. The number can be variable, but very small number of insertions and deletion. Insertion means there is an insertion of a nucleotide, and deletion means there's a deletion of a nucleotide. Then there are copy number variations. Now, copy number variations are actually segments of genes. For instance, segments of the chromosome which are actually amplified or deleted. So if there for example, now if we talk about amplification, there is one chromosome, any chromosome, and there's a region which is like this, and like this. And now there's a small region like this, right? Now this part of the gene gets amplified into multiple copies in the same region. That can be sort of an amplification, for instance, duplication. For example, this region, which you can see here is like this, is multiplied into multiple copies in this way in the same region, and then here there was this point, and then this one. Now here you can see this actually is duplicated. This one copy is duplicated into 1, 2, 3. So this can be a amplification. And if there is a one copy, then you can one copy, a high copy, or multiple copies for amplification. Then there are deletions. So if we say this is an amplification, now if we want to delete something for our understanding from here, we can say that these are the different same genes, then this one, and then this region was deleted. This region is deleted in this. So this is a deletion. So there are different now whole segments of the gene. This can be, these segments can be more than mega base pairs, more than 1 to 100 mega base pairs. So this is different from this one. These are small 1, 2, 3, 4, more than 1 and less than some few, and for few base pairs insertions and deletions. Then these amplifications or deletions can be of whole chromosome level, or even half of the chromosome, or large chromosomal focal regions, or large chromosomal regions. Then there are different variations, large chromosomal variations or translocation. For example, some gene, this gene make a copy and get pasted here, then this gene make a copy and get pasted here. So there are different rearrangements. These are called translocations, right? So there are different types of genomic changes, and as the size increases, just get into the one concept that if as the size increases, for example, this is one nucleotide, then and here it is 1 megabase per region, then as the size increases, and this is the frequency, for example, this is the frequency, this is frequency. So as the size, so now as the size increases, the frequency decreases. For example, this is SNP and this is large chromosomal variation, this one, right? So as the size increases, the frequency decreases, but every type of genomic change has its own importance. For example, this is one example, and it is taken from the TCGA Nature article in 2008. You can see here that different types of mutation can alter pathway activity. Now, these small examples actually I want to share to make you understand that how data gets increased and how data integrates and get complicated. But if you move step by step and try to understand the different data types, then it will be easier for you to go into this data science field. Now, this is example, the different types of mutations can alter pathway activity, right? So one is that if the oncogene gets activated. Now there is another term, oncogene, and then tumor suppressors get deleted, right, or inhibited. So what is an oncogene? Oncogene actually is the gene whose overexpression or overactivation can alter the pathway and normal pathways and make the sample or cell cancerous. And tumor suppressor actually regulates the tumor or cancer. So if the tumor suppressor gets inhibited or deleted or inactivated, it will cause another type of disease. For example, here you can see that activated oncogenes can lead to the whole process. So if this is a TP53 pathway, if you want to study this pathway in detail, you can see that now these are activated oncogenes actually regulates CDKN2A, and CDKN2A is regulating or inhibiting MDM2, and these MDM2 actually are inhibiting this tumor suppressor gene. That is a tumor. So if this MDM2, which is actually regulating the TP53, which is a tumor suppressor, then the overexpression of this gene can inhibit the function of this tumor suppressor. So it means that MDM overexpression is an oncogenic effect, and the inhibition of this is a tumor's inhibition of the tumor suppressor effect. So that may lead to the inhibition of apoptosis. Like this TP53 gene actually do cell apoptosis, and cell apoptosis is very important in cancer cells. So if this gene will not perform cell apoptosis, then it will lead to cell proliferation and further increase in cancer. So now you can see that there is an integrated or combinatorial role of upstream and downstream genes, which can be oncogenic or tumor suppressor. So these are different types of changes. I have explained you in the detail that nucleotide change that can lead to an early stop codon, making a protein non-functional. That can be an inactivating mutation, and then there are types of activating mutations that can make the protein more functional or overactivated. For example, here you can see that at this point, this is a point mutation, like A has been changed to C. Then this is a complete picture of the copy number alterations, as I've shown you the difference between a few insertion deletion and copy number. Copy number is always a big number, starting from a few kilo base pairs to mega base pair, and then 100 mega base pairs until the whole chromosomal region. So the chunks of the genome can be amplified, leading to many copies of an oncogene, which leads to overexpression of the gene. So if the gene is amplified, now you can relate it with the overexpression of the gene, that if the DNA at the DNA level, a gene is amplified and there are many copies of that gene, then that gene will lead to the overexpression of the gene, and overexpression will inhibit the tumor suppressor roles, and then that will lead to the cancer or any disease. And then the chunks of DNA can be lost, and that is one mechanism to lose the tumor suppressor. So if there are genes, for example, if we say that This red represents the tumor suppressor and blue represents the so red represents the deletion and inactivation, and blue represents amplification. So if this whole region in cancer cell is deleted, right, this one, so and there are any tumor suppressor in this gene in this region, then that will lose the important function of tumor regulation and tumor will increase. So there are different types of studies. So you deal with the big data, all the 22,000 genes which are in this picture. You can see that starting from first chromosome to last 23 chromosome and then X and Y chromosomes, they have identified all the deleted regions or amplified regions in this gene. So you can do one by one analysis, or you want to do a cumulative analysis. That depends on your question of interest. So these big data are making things interesting and more conclusive and more relatable towards the improvement of the disease prognosis. Then there is then from mutation to copy number variation, and that from DNA, so that from the DNA we move to RNA, that is called transcription, and the centrodroma, right? So classification of cancer by expression. So there are other studies. Here in DNA we were talking about segmental regions, and now here we are talking about the expression of RNA expression of genes. So RNA expression of genes. So this clustering figure is another example of a big data that was published in 2010. That this is called a heat map. If you can see that heat map. Now they are making comparison of different cancer, even subtypes of cancer have dramatically different gene expression patterns. These represent disease states. So now this is luminal A and luminal B. This is an example of what? Breast cancer. Breast cancer, right? I have a good writing style. Okay, now luminal A and luminal B are different types. Here you can see that now they are trying to explain the big chromosomal data of whole genome in one figure. Now this is the beauty of big data. Now this dark blue and this light blue represents luminal A and B. Probably I don't know what it represents. I have just so here dark represent blue. Now this is a luminal type A, this region, because it's dark blue. This is luminal type B. So now you can see here clearly that luminal type A has a greenish effect. So compared to this red type has a red region. So if this green represents overexpression, then luminal type A has overexpression of these genes, and while these genes are downregulated in red type. So it means that if we know how to arrange the different data types and how to explain different concepts in of RNA expressions using heat map or any other method, then that will help you a lot in finding the solution to your answers. Now, just take an example. If we are a biologist and we just want to explain one gene, how much significant that can be in this era. Today we are dealing with the whole genomes at a time, right? So if we are dealing with whole genomes, then we should, everybody, even bioinformaticians or non-bioinformaticians, everybody should go and they must try to deal with the whole genomes, right? Or transcriptomes or proteomes. Now it's no time, and this can be, these are the big data. Now we don't have time to deal with one gene at a time. So, and for biologists, there are a lot of different tools, like here heat map. Now you can generate heat map using already available commercial tools or even non-commercial tools that will help you. So this whole presentation or discussion is about encouraging everybody to jump into the field of big data. And then there are different, another aspect of example of the you can say understanding the genome. Now we were talking about one gene at a time, then multiple genes, then DNA copy number variations, and then expression, a heat map of the whole genome. Now we're talking about the cancer development. Now this is another process. We have discussed about the evolution term in detail, right? Now there is another example of cancer evolution. For example, if we want to understand the genome, that a normal cell, what was the genome, now at the first mutation type, what was the genome type, and then at the second mutation, third mutation, and malignant cells. So now we can compare, for example, if you want to compare the genome of malignant cell and the early cell of the same individual, we can compare that using the information. Like for example, we can compare the this genome with this genome of the same genome but at different time points. We can understand that when there was a first mutation and when there is a fourth or later mutations or malignant metastatic mutations, what are the difference between the different genomes, and we can understand the evolution of the disease, that how disease is evolved from first mutation to the several mutations. So there are different examples, and there are different ways we can analyze the data that currently in you can say data science. Again, now we have to find this is another aspect. So these are different challenges which we have to which we face, or you can say there are different exciting aspects. But that depends on you, what you want to, how you want to interpret your challenges. For example, genetic alterations is another term. Genetic aberrations, genetic alterations, genetic changes, mutations, whatever you want to say. Now there are number of mutations you can observe. For example, we have a cancer genome and we identified 10,000 mutations, right? Now these 10,000 mutations are very different to analyze. Then we should try to identify, comparing with different databases, that out of these 10,000, how many are functional, or in which pathways, or in which biological processes. So this can be a question, right? Biological processes. Then another question, that or any other gene ontology information, or any other information we want to look for of these functional databases. Then there is another out of those now there's a difference, the difference between being functional and driver in a specific cancer. Now driver, you know what is the difference between driver and my writing is excellent. Well, people should dream of writing like me, right? Because this is the way to go. Okay, now we have discussed the difference between driver mutations and passenger mutations, right? So here you can see that how we can say that these are the drivers. One best aspect is that if we identify the significantly recurrent alteration across the samples, it means that this is another aspect. For example, we want to look for the driver mutations. How we can say that these are drivers? We can just compare those mutations in different samples. For example, we have 100 breast cancer samples. Now if these driver mutations, the mutations which are recurrently altered or recurrently mutated or significantly mutated or are repeatedly mutated, but there are different terms you can see, or you can face in different literature. Recurrent alterations, mutations, significantly mutations. So the genes which are recurrently altered in these 100 breast cancer samples must be drivers, because they are not by chance, they are mutating themselves again and again. So there are so now we are talking about another aspect of big data. One by one we added a lot of things. So this is a whole, you can say, sea. You can dig deep into different aspects, and all these aspects can be analyzed now using different tools which are readily available, freely available on databases. In the next part of the now we have discussed different concepts of disease informatics or cancer informatics. In the next section of this lecture, we will go into the details of different types of databases, different tools available, or different statistical packages you can use to analyze this big data and to conclude that whether which one of the driver mutations, which are the which genes are evolved, how the genome is evolved, or how we can identify expression specific to any subtype, or how which are the tumor suppressors out of the whole genome and oncogenes. So these all questions can be answered in the can be answered by you using different statistical softwares, packages, databases, browsers. So next up we will discuss the different databases. I hope these concepts are clear to you, and I really hope that now you know that why big data is important for every biologist to learn. Thank you very much. Let's meet in the next class. Thank you. Bye bye. Assalamu alaikum.